Hi, I'm Hannah Isabela Ocbina, an incoming second-year dentistry student at the Adventist University of the Philippines. For this video, I will be discussing DNA fingerprinting, an application in biotechnology in the field of medicine. First, what is DNA fingerprinting? DNA fingerprinting, also called DNA profiling or forensic genetics, is a laboratory technique that is used to identify a person based on the unique patterns of their DNA. DNA, or deoxyribonucleic acid, as we all know, is a molecule that carries the genetic makeup of all living things. DNA fingerprinting is used in a variety of situations such as criminal investigations, paternity testing, tracking down blood relatives, looking for cure for diseases, and other forensic purposes. DNA fingerprinting was invented by Sir Alec Jeffreys, a professor and geneticist at the University of Leicester in the UK. Professor Jeffreys, back in the 1980s, was interested in studying the genetic variation between individuals and had done some of the early work to detect genetic differences in humans, since labs around the world are searching through DNA for genes that can cause major illnesses. On September 17, 1984, an accidental discovery had happened. While he was comparing some tiny fragments of human DNA, he found out that there were many parts in the DNA where he could see inherited patterns, but one region appeared to be unique to one individual, which suggested it would be ultimately possible to identify every human being in the world with a unique genetic makeup. And then Jeffries and the other lab workers started to list things about how the accidental discovery could be useful. They gathered samples from all over the lab, smears on equipment, stains on coats, flakes of skins, and rug hairs. And they were able to match each piece of evidence to its owner's unique DNA patterns and decided to name it, thus the name DNA fingerprinting. So for the biotechnology process of DNA fingerprinting, it starts with isolating DNA from any part of the body. It can come from a swab inside your mouth, from your skin, the roots of your hair, your saliva, sweat, blood, or other body fluids. Next, it will undergo the process called PCR, or polymerase chain reaction, which is used to make millions of copies of the DNA regions. After the DNA is isolated and more copies of the DNA have been made, the DNA will be tested. The scientist will treat DNA with restriction enzymes, which will produce different sized fragments, which are known as restriction fragment length polymorphisms, or RFLPs. These fragments will then undergo gel electrophoresis, wherein an electrical current is applied to a gel mixture, which includes the samples of the DNA. The electric current causes the DNA strands to move through the gel, which separates the molecules of different sizes. The DNA is then cross-linked against the nylon using heat or a UV light. The probe shows up on photographic film as the strands of DNA decay and gives off light. In the end, it leaves dark spots on the film, which are also known as the DNA bands of a person. Once the filter is exposed to the X-ray film, the radioactive DNA sequences are shown and can be seen with the naked eye. This creates a banding pattern, or what we know as DNA fingerprints. For crime investigation, as we can see in the picture, the sample from the crime scene perfectly matches the sample coming from suspect 2, which guarantees that person is the primary suspect. So for the advantages and disadvantages of DNA fingerprinting, for the advantages, first, it is simple and less intrusive. Second, collected evidence can be stored indefinitely. DNA fingerprints can be stored for a long time. Hence, they are easily accessible for analysis. Third, it can reduce innocent convictions. Fourth, it can be used to identify hereditary diseases. For the disadvantages, First, it can be a violation of one's privacy. Second is errors in performing the technique or improper handling of the sample that may produce incorrect results. Third is hacking can become a major concern. Fourth is DNA information could be misused to discriminate against an individual based on their race, ethnicity, origin, or health risks. So that's all. I hope you learned something from this video. Bye!